Hello everyone, this is Sean Taylor, Field Application Scientist Manager for Bio-Rad Canada, and I'd like to go over how to use the image tools that appear above an image that is acquired with any of our blot or gel imaging solutions from Bio-Rad. This is using ImageLab software, which drives all of these solutions. So if we start here with the Image Info tool, we see that this tool contains all the metadata from an image, including the imager, the exposure time that was used, the serial number of the imager, specific applications that were chosen when the image was assessed, excitation source, date, and a variety of other information around the resolution of the image. A person can also add notes about the experiment, so this is quite useful to assure that the data from an image is logged so that there isn't just a single file name saved, but some descriptive data about how that experiment was chosen, and all of this resides with the image file itself. This is the three-dimensional viewer, which is quite a useful tool when looking at an image and wanting to just by eye assess the densitometric differences between the various bands. So you can see here what we've done is we've converted a flat image of lanes and bands to a three-dimensional image where we still have the lanes and the bands appear as peaks of density. So you see the lane width and the bandwidth, and I can make adjustments with this tool at the bottom here. So we can see the size of the densitometric peak that rises above the image. These three-dimensional peaks are generated by a camera which is situated above the image, which views the light coming from, for example, a chemiluminescent or UV fluorescent blot or gel coming towards the camera. So the camera sees light as different intensities, which is exactly what we would like the camera to assess when we're doing densitometric analysis of lanes and the individual bands within those lanes. So what the user needs to do in our software, in ImageLab software, is define the lane width and, in this dimension, the bandwidth, and therefore the software will densitometrically analyze the volume of this three-dimensional peak that rises out, which would be the volume of the light intensity that comes from that band. This next tool is used to change image colors. You can click on this tool and alter the color of an image, but bear in mind that the camera is a grayscale camera measuring shades of gray of an image. So all of these different color settings that you can choose to convert an image to make it look like a Kumasi blue gel, for example, are artificial colors that are applied by the software. Now, of course, the densitometric data doesn't change in the image, but it would likely be preferable from a journal to receive the image itself in the native color settings of the camera. And given that all the cameras are grayscale, and purposely grayscale so that they can measure the shades of density of bands, it's probably preferable to leave images in a grayscale format for publication. This is the Image Transform tool. This tool permits the user to change the brightness and contrast of an image. All of these settings have no impact, again, on the raw densitometric data of these bands. No matter how these settings are changed, the densitometric volume of those peaks remains exactly the same. These settings are strictly used to adjust the brightness and contrast to the tastes of the user for publication purposes. This tool is to center the image, and this tool is to zoom out and zoom in, or again, we can go back to centering the image in the frame. This final tool, display gel options, I'll discuss in a later video that will go over how to use our software to densitometrically analyze the lanes and bands within those lanes.